Ben Turner from the Smithsonian Tropical Research Institute in Panama, outside of Panama City. So he's traveled relatively long way to get here. Ben's a staff scientist, works on all sorts of aspects of tropical soils and soils in general, and I'll let him tell you all about the ecology of tropical forests.
Well, good morning, everybody, and welcome. Thanks for coming to this early session. If you were at the party last night, thanks for coming with your hangover. I hope it's not too bad. It's my pleasure to be here to give this talk. I always enjoy coming to Canada, and I want to thank Carl and Joanne Whalen and the other organisers for giving me the opportunity to be part of your meeting. As Carl said, I'm a staff scientist at the Smithsonian Institution based in Washington, D.C. Just in case there's any booing, I wasn't quite sure. Of course, you're all too polite. Canadians are too polite to boo. We're actually located in Panama City. We've been there about 100 years, associated with the Panama Canal. There's a group of about 30 scientists there working on all different aspects of tropical biology. And we host also more than 1,000 scientific visitors per year, many from Canadian institutions. And my role there in Panama is soils, is the study of soils. And my talk today will be about how soils interact with geology and with climate to shape the ecology of these fascinating ecosystems. And so, for many temperate ecologists, and perhaps also many temperate soil scientists, there's a really strong perception of what a typical tropical forest soil looks like. And perhaps something like this. This is an oxysol, strongly weathered oxysol in one of our key research sites in Panama. If you're interested in the US soil taxonomic classification, as I'm sure many of you are, it's a candy eudalphic eutrodox, deep, strongly weathered, high in iron oxides, giving this really vibrant red colour and very infertile. A classic tropical forest soil. But actually, as you may not be surprised to know, there's enormous diversity of soils in tropical forests, and I'm just showing here some examples of soil profiles from our long-term research sites in the neotropics along the top panel in the Americas, and in the Asian tropics along the bottom there. And what I hope you can see is that, in fact, tropical soils vary markedly. We have an enormous range of soils at the order level, at the highest level of soil classification, from, yes, strongly weathered oxysol, for example, there in Puerto Rico and other places in South America, but also there's albisols, altisols, and even mollisols, the soils of the prairies, which I'm sure many of you are very familiar with. We find them in lowland tropical forests. So a real diversity of soils in the tropics. Of course, people are not particularly interested in the diversity of soils in tropical forests. They're interested in the diversity of organisms, of the plants and insects and animals which make up these incredibly complex ecosystems. And this is our primary research site in Panama. This is Barro Colorado Island. It's an island formed in the middle of the canal by the flooding of Gatun Lake. And so the canal runs from the top right of the image there across towards the top left. The boats go through there from the Pacific side through to the Caribbean. And this island, about 1,800 hectares, was given over to the Smithsonian for study in the early part of the 1900s, and is probably the most studied piece of tropical forest on the planet. Now we have a long-term forest dynamics plot on the top of this island. It's 50 hectares in size, 1 kilometre by half a kilometre. And in that plot we measure every 5 years, every stem bigger than 1 centimetre diameter at breast height. So there are about 200,000 trees in this plot. And within those 200,000 trees, there are about 300 species, 300 tree species in 50 hectares. And that's not including things like lianas, the climbing plants, which add about another 150 species. So incredible diversity in a relatively small area of forest. And we have many of these plots around the world in temperate, in tropical and temperate forests. And I'm showing them here on the map of the world. You can see many sites in Southeast Asia, many sites in the Americas. There's even 2 plots in Canada. One up in Scotty Creek, which is in the Northwest Territories, a boreal forest site, and also a site north of Toronto, not too far from here, called Halliburton Forest. And so in these plots, they're typically 25 or 50 hectares in size. They're all monitored and measured in exactly the same way as the plot on BCI, and this gives us the opportunity to examine and to understand patterns of tree diversity at the global scale. And what we find is that actually the plot on Barra, Colorado Island is not that diverse for a tropical forest, even though there are 300 species. So, for example, if we go to a site called Lambir Hills, which is in Sarawak, on the island of Borneo, in a plot almost the same size as on Barra, Colorado Island in Panama, we find more than 1,000 species of tree. And if you're wondering about the Amazon, which we often think of as being one of the most diverse places on the planet, we have a plot in Yasuni National Park in Ecuador, which also has more than 1,000 species of tree in only 25 hectares. And in fact, there are some single hectares of the plot in Yasuni that contain more than 600 species of woody stems. So an incredible diversity. And in fact, there's a new plot which is almost finished in Manaus in the central Amazon, which is 25 hectares in size and looks like it's going to have about 1,400 species of trees. So a remarkable diversity in these forests. And if we compare that to comparable size plots in temperate forests, you can see this remarkable difference. We have I've put 2 examples up there, Whiten Woods in the UK, with 23 species, and the Halliburton Forest north of Toronto here, beautiful forest, but only 30 tree species. So this is not to denigrate temperate diversity, but just to demonstrate the real challenge that we have as soil scientists trying to understand the influence and the impact of soils on plant distributions and plant ecology in tropical forests. Effectively, we have the entire temperate tree flora in the space of a few football fields in these forests. And so diversity shapes many of the big questions that we're interested in at my institute. 3 of those I'm going to mention here. First, of course, why are these forests so diverse? How can we explain this remarkable plant diversity? What's creating this diversity and what's maintaining this diversity? What's allowing so many species to share this space, this ecological space? We're very interested in patterns of distribution of tree species. Why do tropical tree species grow where they do? And finally, something that's becoming more and more emphasised as modelers try to incorporate tropical forests into these big Earth system models to predict the future climate, is what controls growth in tropical forests. We know that carbon dioxide concentrations are increasing in the atmosphere, and this perhaps could fertilise tropical forests, causing them to grow faster and therefore sequester some of this excess carbon. But perhaps these forests are limited by nutrients, and this predicted sink doesn't exist. So we're very interested in trying to understand what controls growth of tropical trees. And so my research as a soil scientist in Panama is to bring a soil perspective to each one of these questions. And I'm going to structure my talk around these 3 questions, showing, I hope, how I think that soils influence and are involved in answering some of these most important questions about tropical diversity. So my first question is, how do soils influence plant diversity? And to do this, to answer this question, I want to take you back to some basic soil biogeochemical theory. I want to talk about soil forming factors. And in fact, as all good soil scientists know, there are 5 soil forming factors shown here, which interact to give you the soil in any one particular place. These are climate, organisms, relief or topography, parent material, and of course, time. I want to zoom in and focus on time. In particular, I want to look at how time influences nutrient availability and dynamics in ecosystems. And so when we start off with a new soil, maybe it's a volcanic flow or a sand dune or a glacial moraine, typically that soil has very little nitrogen, and that's depicted here on this schematic diagram. We've got nutrient content on the y-axis and time on the x-axis. Time could be hundreds of years, it could be thousands of years, it could be millions of years, depending on those other soil-forming factors. And so nitrogen starts off, as you can see, at time 0, very low in most soils, but it accumulates. Organisms bring nitrogen into the system, they fix nitrogen, and the nitrogen capital of the soil increases through time. Phosphorus follows a very different pattern. Typically, soils have about as much phosphorus as they will ever contain when they start to form. So you can see there at time 0, the maximum total phosphorus content. And then over time, phosphorus is gradually leached and eroded from the soil, and the phosphorus capital of the soil declines. As you can see there, as soils age, phosphorus concentrations decline to relatively low levels. And this has important ecological consequences. What we typically find is that on young soils, primary productivity is limited by nitrogen availability. Then the nitrogen accumulates, phosphorus is declining. We go through a period of co-limitation, of nitrogen and phosphorus co-limitation. And eventually, on stable land surfaces, phosphorus declines to such an extent that we start to see phosphorus limiting primary productivity. And this strengthens as the ecosystem develops further. And in fact, it's not just the loss of total phosphorus that is driving this pattern. We also see strong chemical changes of phosphorus in the soil profile. And this is a classic model of phosphorus transformations during pedogenesis, developed in New Zealand. And what it's showing there is you can see the same total phosphorus decline as in my previous figure, but now we can see variation in the chemical forms of phosphorus in this soil. And so the blue panel, the blue area, is the primary mineral phosphate, the apatites that are in the soil originally, in the original parent material. This declines very quickly as soils acidify during pedogenesis. We accumulate organic phosphorus, that's the green area at the bottom, as organisms incorporate phosphorus into their bodies and then return them when they die to the soil. And we also get this occluded pool of phosphorus, what we call occluded phosphorus. This is phosphorus that gets incorporated into secondary minerals, iron and aluminium oxides, and this accumulates as soils develop. And so you can see that by the time we get to stable, old land surfaces, most of the phosphorus, or all of the phosphorus, is in these two forms, in these organic, bound up in organic forms, and also in these secondary mineral forms, which we typically consider to be poorly available to plants. So there's two forces acting to drive phosphorus limitation on old land surfaces, this decline in total phosphorus and also chemical changes in the nature of phosphorus in the soils. Now we can examine these processes, of course, although they're happening over many thousands or millions of years. We can use space-for-time substitutions by examining soil chronosequences. And this is an example, actually one of my favourite examples of a soil chronosequence. It's on the west coast of New Zealand, near a place called Haast. And what's happening here is we've got the Tasman Sea on the upper right of the image. On the left, we've got the Southern Alps, this mountain chain running through the spine of the South Island of New Zealand. And about every 300 years, there's an enormous magnitude 8 or more earthquake along the Alpine Fault, which runs across the top left of the image there. This causes uplift, landslides, river terraces get moved around. And we end up with an enormous pulse of sediment flowing into the Tasman Sea through the Hass River at the top of the image there. And this sediment washes back by longshore drift as a dune. So each one of these parallel ridges that you can see in the image is a dune that represents a unique earthquake event. And so we can go back from the right of the image, the youngest dunes. We can go back through time, examining the soils, looking at the forest, and we can reconstruct how the ecosystem has changed, in this case over about 6,500 years. And interestingly, the last earthquake, the last major earthquake along this fault was in 1717 AD. So they're absolutely due another one any day. They seem very blasé about it at the moment, but we know it's coming. So these changes in the soils have an important effect on the tree community. I mentioned how this influences nutrient limitation and changes from nitrogen to phosphorus limitation during ecosystem development. We also see changes in a biomass of forests. So this is an example from the east coast of Australia, at a place called Kalula. It's another dune, a dune sequence formed during interglacial cycles, moving sand around on the east coast of Australia. What you can see on the left of the image is a young dune, about 1,000 years old. You can see in the lower part of that panel, the soil is formed in relatively fresh sand, this yellow-coloured sand, and the forest there is dominated by plants which fix nitrogen. It's fairly low stature, and this is a typical example of a nitrogen-limited stage of the ecosystem. We call this the progressive developmental stage of the ecosystem. After about 200,000 years, the soils look like they do in the middle panel there. What's happened is that we've started podzolisation. Iron oxides have been leached from the upper part of the soil profile by organic acids from plant litter inputs. And so the iron oxides are accumulating in a spodtic horizon about 1 metre down in the soil, leaving this bleached quartz E horizon. But at this point we've got this kind of co-limitation going on, but there's plenty of nitrogen now in the system, there's still some phosphorus available to the plants, and we're in what we call the maximum biomass phase. You can see big eucalyptus trees there in the top part of that central panel. This is what you may remember from geography classes at school as the climax vegetation. But in fact, on stable surfaces, these ecosystems continue to develop, and they go into what we call a retrogressive phase. And you can see on the right of the image that this podzolisation now has moved this B horizon, this spodtic horizon, down to about 20 metres below the soil surface, well out of reach of plant roots. The soils are incredibly infertile. They contain less than 10 parts per million of total phosphorus, super infertile soils. And the plant communities there are stunted, short stature. If you were standing in this forest, those trees would be about up to your head height. Low stature forest, strongly limited by phosphorus availability. So there's major changes in the biomass of these forests related to these changes in nutrients that we see through time. Now, we see very similar patterns in the soils and in the plant communities wherever we find a long-term retrogressive chronosequences, and I'm showing here some of the best well-studied examples around the world. In fact, there are not many well-studied retrogressive chronosequences. There's let's have a look. Well, there's the Franz Josef glacial chronosequence in New Zealand. This is a post-glacial sequence. Another famous one is at Glacier Bay in Alaska. There's volcanic sequences, for example, the iconic Hawaiian Islands chronosequence that Peter Matusek and his colleagues have done so much work on. There's the Hass sequence in the bottom right there. All following very similar patterns of soil development and of tree community progression along these chronosequences. But I started off this section of the talk asking about diversity. And to address this question, I want to take you to one particular chronosequence, which is the Joring Bay chronosequence in Western Australia, the centre of this panel here. Now, it doesn't look much, but it's actually a remarkable place to do soils work, because it's in a global biodiversity hotspot. And so it's a hotspot for both species richness and endemism. There are many species there which occur nowhere else. And what we find in this area is very clear 2 million year old chronosequences all the way up the coast of Western Australia. And these are formed by glacial interglacial cycles. So as sea level drops during glacial cycles and then comes back, rebounds after as the glaciers melt, this pushes up debris, sand onto the west coast of Australia, forming long linear dune ridges all the way for hundreds of kilometres up the coast. So we can go to these dunes and reconstruct a chronosequence stretching all the way back into the early Pleistocene. And because it's in a global diversity hotspot, we can start to ask questions about how soil development is driving or affecting or influencing the diversity of plant communities. So just to show you very quickly how the soils progress, I often talk at ecology meetings, and the ecologists are not particularly interested in looking at soil profiles, so I wanted to take advantage of being here at the soils meeting to talk a little bit about the soils. And so what you can see on the left of this panel is a young soil. It's about 1,000 years old. It's representing the Holocene dunes. It's formed in carbonate sand, so there's relatively high pH, high phosphorus, and plants growing on these carbonate sands seem to be limited by nitrogen availability. And what happens is as the soils develop and acidify, that carbonate is leached from the profile, forms a petrocalcic horizon at depth in the soil, and we're left with soils typical of this middle panel here, these very bright orange sand profiles. This is the residual iron oxide still left on the sand grains after the carbonate has been leached. We've lost much of the phosphorus with the carbonate, but there's still moderate amounts of phosphorus there. And these soils represent the middle Pleistocene dunes between 100 and 500,000 years old. And then by the time we get to the very old dunes, these early Pleistocene, perhaps even Pliocene, late Pliocene dunes, we've lost all those iron oxides off the coatings of the sand grains through leached away with organic matter. We're left with these bleached quartz sand profiles, and almost all the phosphorus is gone, so now we're left with some of the lowest phosphorus soils that you'll find anywhere in the world, super low phosphorus soils, and strong phosphorus limitation of plant communities. So, if we look at plant diversity along these 2 million year chronosequences, we find very striking patterns. And I'm showing here along the bottom panel there 3 different measures of diversity, of plant diversity. If you look on the left, the lower left panel there, this is the red circles are alpha diversity. This is the number of species that we find in a plot. We typically use 10 metre by 10 metre plots on these dunes. So if you go to a plot, this is how the diversity looks. You can see increasing throughout the system. If we go to the gamma diversity, which is basically adding up all the species we find in the various plots on a given dune system, very clear patterns of increasing plant diversity through time. And if we look at beta diversity, the right-hand panels, this is beta diversity for all plants in the middle panel, and then the woody plant beta diversity on the right. You can see again very strong patterns of increasing diversity as soils age and as phosphorus declines along these chronosequences. And it turns out that wherever we find long-term retrogressive chronosequences around the world, that the patterns of diversity are the same, whether it's on Swedish islands, sand dune systems, post-glacial marine surfaces in New Zealand, Glacier Bay in Alaska, even the Hawaiian Islands. We find this pattern of increasing diversity, so soil development somehow driving increasing plant diversity on old soils. And we spent a long time thinking about this and trying to understand what could be driving this pattern. And we came up with this conceptual model to describe how we might go from pedogenic stage, from weathering stage of soils, pedogenesis basically, to local plant diversity. And we separated possible explanations into two groups. You can see the yellow box there is what we call local processes. So things which might be driving or maintaining diversity at a given at that local scale. This could be the diversity of nutrient forms, for example. So plants perhaps share a limiting resource by using different forms of it, for example, perhaps using different forms of organic phosphorus in soils. Perhaps spatial heterogeneity increases as soils get older, and so there are more niches to support a greater diversity of plant species on older soils. So all these things acting at local scales and have been hypothesized to be important in driving patterns of diversity along resource gradients. But there are also a bunch of processes which we call regional processes, which could include simple abiotic variation in abiotic conditions, perhaps commonness of habitat, so the most common regional habitat providing more species at certain stages of the system. And we call these regional processes or things which are acting as environmental filters. And we expected, when we started studying this and trying to add in information on each of these, to test each of these hypotheses, we expected strong effects of phosphorus. We knew that phosphorus was declining very strongly along these chronosequences. And we also knew that plant nutrient acquisition strategies were diversifying along this chronosequence. So, for example, I'm showing here some data on mycorrhizal or root nutrient acquisition strategies. And we find that on the oldest soils, on the right of those panels, we start to see ericoid species. We start to see species with cluster roots, which you can see an example of in the bottom right. These are roots. These formed by this plant family, the Proteaceae, very small, like little bottle brushes, which exude enormous concentrations of organic anions to mine small concentrations of phosphorus from very infertile soils. And so we know that phosphorus is important and becoming increasingly important to the plant communities on these old soils, and we expected that this would probably be driving these patterns of diversity. And so we gradually populated that conceptual model, and this doesn't look much. It looks very complicated, but it's the end point of an enormous amount of work testing each one of these models one by one to try and explain how we get from weathering stage, from pedogenic stage to local plant species richness. And so each one of these arrows, this is a structural equation model. It tests every possible pathway through these various models, and you can see chronosequence stage at the top and plant species richness at the bottom, local diversity at the bottom. And yes, total phosphorus does have enormous explanatory power at the local scale. And so we see these arrows, the size of the arrows you can think of as the kind of strength of the relationship. And so yes, as we go through across these long time scales, total phosphorus changes, and it influences things like spatial heterogeneity in the soil, diversity of phosphorus forms, all these things which we think of as affecting, perhaps maintaining local plant diversity. But these things have very little explanatory power across the chronosequence as a whole, and in fact, the only explanatory variable linking chronosequence stage pedogenesis to plant diversity was pH. The most straightforward measurement that we make on soils. And so the conclusion is that this long-term weathering of soils, pedogenesis, reducing plant diversity, reducing pH is driving plant diversity. And it makes sense when we think about the regional species pool. And so these chronosequences are located in Western Australia. It's one of the most stable pieces of the planet. It's not been glaciated since the Permian 250 million years ago. It's incredibly stable, and you can understand that most of the plant species in this region are very well adapted. They've evolved in the context of infertility, of acidic soils, strongly weathered soils. And And so, in fact, as we go along that chronosequence, the regional pool, there are many more species in the regional pool which can populate the older stages of the chronosequence. So it's all about this regional pool and how environmental filtering is driving distributions of diversity across this chronosequence, and how soil development is therefore driving these patterns of diversity. I'm going to move on to my second question. And finally, I'll get to Panama, to the tropics. The question is, how do soils influence the distribution of tree species in these very diverse tropical forests? And I want to take you to Panama. There's Panama. It's right down there at the bottom of North America, bridging the continents, bridging North and South America. It was incredibly important in the relatively recent geological past. It formed a barrier to ocean currents when it formed about 3 million years ago, blocking the flow of water from the Pacific into the Atlantic, changing the Caribbean forever. It allowed animals to interact, North and South American animals to interact, and of course all the North American ones take over, as seems to be the way. It's also very diverse topographically, as you can see there, and it's very diverse geologically, and this is a real strength when we want to look at the interactions of soils and plant communities. So I'm going to zoom in on this area here. This is the central Panama area and the Panama Canal. What you can see here is an image showing very clearly the legacy of one of the legacies of the American involvement in the Panama Canal. And so the canal is running from the Pacific Ocean on the bottom right of the image through Panama towards the northwest, which is counterintuitive. Most people expect that the canal runs east-west. Actually, it runs south-east to northwest. So running through there into Gatun Lake, you can see Barra Colorado Island is right there in the middle, and then it exits into the Caribbean to the top left of that image. So the boats go through there. You can see Panama City in the bottom right as well, where my institute is based. And what you can see is this swathe of intact tropical forest running around 10 kilometers around the canal. And so the Americans have this zone around the canal very well protected, and it's remained so to this day. And what we have across this 50 miles of the isthmus is a very strong rainfall gradient. So rainfall is fairly low, which you might not think is that low, but it's less than 2,000 millimeters on the Pacific coast there, actually down to about 1,600 millimeters just to the south of Panama City. And then it increases to more than 4 meters per year on the Caribbean coast. So a fairly strong rainfall gradient spanning this 50 miles of the isthmus. But also in this area, the soils, the geology is very complicated. So there's the geology map of this same region. It's a real mixture of volcanic and sedimentary bedrock. There's limestone in there, and so that yields a really complicated range of soils. And I've put some of some soil properties up there on the right. You can see an enormous range of phosphorus and pH, a particularly plant available phosphorus, which varies about 300-fold in this region. So we've been using this area as a framework for understanding distributions of plant species in relation to environmental variables. And we've known for a fairly long time that moisture is very important in structuring these very diverse forests. We have about 1,000 tree species in our plot network, which you can see in the top right panel there. About 1,000 species of trees which we're monitoring in these plots. And they definitely respond to moisture. So this is an example for one tree species, which is a Xylopia macrantha. It's in the custard apple family. There's a picture of the fruits and the leaves down there. And so this is an example of a relatively drought-sensitive species. And so what you can see in the top right panel is our plot network. All the coloured or shaded circles are where this Xylopia species occurs, and the open circles are where this species does not occur. And what you can see is that it's almost always present on the wet Caribbean slopes of the isthmus, but it's absent on the drier Pacific slopes. And if we go to our 50-hectare forest dynamics plot on BCI, which is approximately in the middle of this gradient shown by the square there, each one of these points on that map, this is 1 kilometre by half a kilometre on the plot. Every point on that map is an individual of this species, and you can see that it tends to occur on the slopes in the south and the southeast part of the plot, and they're areas which we typically consider to be relatively moist in the dry season. And so an example of a drought-sensitive species, and we can do this for many of these 1,000 species in this area. So we know moisture has a really strong influence on species distribution. What we've been able to do more recently is to add soils and a soil explanation to this understanding. And so I'm showing you 3 examples here of individual species responses to phosphorus. And so if you look at the top panel, this is an example of a tree which is Cavanalesia platanifolia. I'll show you a picture of it in a minute. It's an iconic Panamanian canopy emergent tree. And what I plotted here is the probability that this tree occurs in a plot on the y-axis against resin phosphate. This is a measure of readily exchangeable phosphorus in the soil. And what you can see is that Cavanalesia basically has a very strong affinity for phosphorus. It almost always occurs at high phosphorus, and it never occurs at low phosphorus. And we take the first order parameter of this model, which is plotted there through the distribution, and we call this the effect size for phosphorus. So Cavanalesia has a very strong phosphorus affinity. You're almost guaranteed to find it in high phosphorus, and it has a phosphorus effect size of +2. Now the middle panel shows another species, Eschwellera pitierii. Eschwellera is a very common genus all across the Amazon basin. And in Panama, this particular species has a very negative phosphorus association. And what you can see is that at low phosphorus on the left of the image, Eschwellera tends to be present. You're almost guaranteed to find Eschwellera in a low phosphorus plot. But if you go to high phosphorus, the right side of the image, you'll never find Eschwellera. It's excluded from high phosphorus sites. And so it has an effect size of -2 for phosphorus. In the lower panel, I'm showing a species which has basically no affinity to phosphorus. This is Inga vera. It's a nitrogen-fixing legume. Inga is a very speciose genus in Panama. There are many species of Inga. This particular one has a very strong moisture association. It only occurs at low moisture, but it doesn't care about phosphorus. It's a phosphorus generalist, and it has an effect size close to 0. And we can do this for every species in the canal area in our plots and for every soil nutrient. And so we can generate effect sizes for moisture, for phosphorus, for calcium, and for other soil properties. And it turns out that, in fact, before I get into that, let me just show you a picture of Cavanalesia. There it is. This is if ever you go to Panama and you want to take a trip to the beach, the Pan American Highway runs west out of Panama City, and they call this the last tree before the beach. It's right back next to the road. Beautiful canopy emergence. They're dry season deciduous trees. And on the right, I'm showing, this is a hillside on the banks of the Panama Canal. And what you can see on the right of the image is these dry season deciduous Cavanalesia covering the hillside. And on the left of the image, no Cavanalesia, not a single Cavanalesia. And in fact, this is a geological fault running up through the banks of the Panama Canal. On the left of the image, we've got basaltic soils, very low phosphorus, no Cavanalesia. And on the right, we've got marine sedimentary soils, very high phosphorus and abundant Cavanalesia. So a real striking visual example of the faithfulness of these species to their phosphorus affinities. And so, as I mentioned, we can do these analyses for many of the species, for all the species in the plot network. And it turns out that, in fact, we can explain the distributions of the majority of the species in our plots based on just 2 variables, just moisture and phosphorus. And plotted here is the effect sizes for moisture and phosphorus for a 200 or so of the most common, the most abundant species. And so we can explain now the distributions of almost all. There's only about 30 or 40 species in the network which don't have strong affinities to one or other of these properties. So we can explain the distributions of these very diverse plant communities based on simply on these two key variables. And there seems to be implications for understanding the evolution of plant species in these forests. And I'm showing here two species in the genus Tababuia, which have very different phosphorus affinities. On the left, you can see Tababuia guayacan. This is a very well-known tree in Panama. Flowers, these beautiful yellow flowers at certain times of the year. Tababuia guayacan is a low phosphorus specialist. It only occurs at low phosphorus. And on the right, I'm showing Tababuia rosea. This is so same genus, but a very different phosphorus affinity. This species only occurs at very high phosphorus. And so this is fairly common. We have many of these congeneric pairs of species in the region, and they tend to have very different moisture or phosphorus affinities. And so this suggests that in fact soils or differences in soils, differences in nutrient availability, differences in climate are driving speciation. They're providing the space for speciation to take hold in these forests. Okay, I'm going to move on to my final question, which is, does phosphorus limit tree growth in lowland tropical forests? And I've talked a lot about phosphorus limiting productivity on old soils, so you may think this should be straightforward, but in fact, it's far from straightforward. We do expect phosphorus limitation in tropical forests. It's one of the strongest paradigms in ecology is that tropical forests are limited by phosphorus availability, but it's almost all based on theory. So we know that many of the soils are strongly weathered, although as I showed you earlier, not all of them. There's typically relatively low available phosphate in the soils, and when we look in leaves, we find high nitrogen to phosphorus ratios, suggesting phosphorus limitation. We know that nitrogen is fairly abundant in tropical forests. Nitrogen concentrations in the soil are high, nitrogen mineralization rates are very high. We know that nitrogen deposition is increasing in the tropics. 15N values in leaves are also very high, indicating an open nitrogen cycle. But we do have this paradox of legume abundance. About 10% typically of the stems in many tropical forests are legumes. They're not always fixing nitrogen, but nevertheless, presumably they have an advantage. But actually, experimental studies to demonstrate the nature of nutrient limitation in tropical forests are equivocal. In fact, there are relatively few experimental studies of nutrient limitation in tropical forests, but they tend to show very few responses to the addition of any nutrient. And in fact, the only experimental study in a lowland tropical forest that has produced a response to nutrient addition is in the Hawaiian Islands. Work by Peter Matusek and colleagues has shown very clearly that on the older Hawaiian Islands, about 4 million years old, there the tree community responds to phosphorus addition. But on Hawaii, you remember, there's only one species of tree, Metrosideros polymorpha. In contrast, in species-rich lowland tropical forests, there's only been a handful of studies, but they've all found no growth response to phosphorus addition. So we're left with this kind of paradox, which is theoretically we expect very strong phosphorus limitation of tree communities in the tropics, but experimentally we find basically no evidence in support of this paradigm. Now we have a fertilization experiment in Panama. It's been running for, well, actually it's the 21st year of nutrient addition this year, so it's it's the longest nutrient addition experiment in tropical forests anywhere in the world. This is a map of the plots. It's about a 30-hectare area with N, P, and K added in factorial combinations to 40 by 40 meter plots. And we measure all sorts of variables in this forest, including trunk growth, litter fall, root production, lots of microbial properties as well. So 20 years of nutrient addition, 20 years of phosphorus addition to this forest, but there's a very short story here, which is that at least after 15 years and now after 20 years, we find absolutely no change in the growth of trees in this tropical forest in response to 15 years of phosphorus addition. And the phosphorus availability in the soil has really increased significantly. It's now higher than any soil in the region. But absolutely no effect of any nutrient on growth. So really questioning, are these forests really limited by nutrients? So to address this, we took a different approach to try and understand nutrient limitation in these forests. And we used the phosphorus gradient across the Isthmus of Panama that I mentioned already to look at growth rates in these plots. And I'm sure you're all looking at this forest and thinking, Tababuya Guayacan, this is a low phosphorus part of the canal. And if you were thinking that, you're absolutely correct. There's Tababuya Guayacan flowering on the banks of the canal on very low phosphorus basaltic soil. So we can go to plots in our network across the canal and measure growth and use the natural phosphorus gradient to perhaps try and see growth responses to phosphorus or moisture or any nutrient. And in fact, what we find is that the plants are limited by phosphorus. Species are limited by phosphorus, but at the species level. So this is the results of a hierarchical linear mixed effects modelling exercise. About 20,000 individual trees went into this model in about 500 species. And what I'm showing is on the left-hand side, this is growth responses, diameter growth per year, and responses to phosphorus for large trees in the top and for small trees in the bottom. And on the right, I'm showing moisture, responses to moisture, again for large trees at the top and small trees at the bottom. So if you look at the top left panel, this is the response of large trees to phosphorus. The black line is the average species response, if you like. It's the accumulation of responses of these 500 species. The average response, you can think of it as the average species, if you like, response to phosphorus. And in fact, the individual species responses are shown in faint grey. You may not be able to see those. But what we find is a very clear, relatively small, but very clear response to phosphorus. So higher phosphorus trees are growing faster. Individual species are growing faster. We find the same pattern for small stems. You can see in the bottom left panel, species are growing faster with more phosphorus. Moisture has kind of equivocal effects. There's no effect for large trees of changing moisture, in this case modelled as dry season moisture deficit, so in the top right panel. And if you look in the bottom right panel, for small trees there is a strong effect of moisture. Small trees are growing faster in wetter sites, and we attribute this to small trees having shallower root systems and responding therefore to moisture. So we've got individual species. At the individual species level, the trees are growing faster with more phosphorus. But there are two unusual things happening. This is a plot of the observed growth, the actual growth rates of every individual stem in our plots, plotted against the phosphorus affinity of the trees. So if you remember, the phosphorus affinity is a value between about positive and negative 2 that reflects the distribution of the species in the environment, so whether it prefers or whether it grows at high or low phosphorus. So when we plot I should say that the small black points are individual stems, the blue circles are the species mean growth across the plot network. And so when we plot these against these growth rates against phosphorus affinity, we find something very unusual, which is that the low phosphorus species, these species which grow exclusively on very low phosphorus soils, in fact, there's a lot of variation as you would expect from these very complicated forests. But actually, the lowest phosphorus species are growing the fastest of the entire community. So you can see this weak but significant negative response or negative correlation between observed growth, and species phosphorus affinity. So although individual species are growing faster with more phosphorus, in fact the lowest phosphorus species are growing much faster than species which have high phosphorus affinities. So something unusual is happening. And in fact, when we look at the community level, when we add up the growth in a plot, in a 1 hectare plot, all the individual stems plotted here. Again, there's individual points, the small black points are individual stems, and the red circles this time are the community mean, so the mean in a plot, plotted against the resin phosphorus. We find no effect, no response to resin phosphorus. So we're left with this, this kind of paradoxical, these paradoxical findings, which is that individual species respond to phosphorus. Low phosphorus species are growing the fastest, but somehow, because these species turn over across the phosphorus gradient, they occupy narrow parts of the gradient, that the turnover of these species means that at the community level, somehow the growth equalizes out, and so across this 300-fold phosphorus gradient in the region, we find absolutely no variation in community level growth. And so this perhaps explains why experimental studies at a single site working at the community level have failed to find consistent responses to phosphorus availability. Okay, that's the end of my talk. I just wanted to quickly summarize and remind you about my points. I showed you that pedogenesis, soil development, drives patterns of plant diversity across long timescales. The greatest diversity occurring on the oldest soils in ecosystems worldwide. A real role for soils in understanding species diversity. I showed you that species distributions in these very complicated tropical forests with many hundreds of species in small areas can be explained quite simply by two key variables, by moisture and by phosphorus, allowing us for the first time to start to understand how these complicated tree communities are assembled. And finally, I showed you that understanding tree growth in response to nutrients in tropical forests requires that we examine patterns at the species level, at the level of individual species, and the turnover of species across fertility gradients allows tree communities to equalize their growth across very strong gradients of soil fertility. Okay, and I'll leave it there. Thank you very much for your attention, and I'll be happy to take questions if there's time.
I think we do have time for one or two questions for sure.
I've got a question. Did you ever look at root morphology of these different trees? Is that what is causing their response to phosphorus oxidation?
The question is, have we looked at root morphology, perhaps root traits? We are, yes, we're starting to look at this. There is a lot of variation. There's a lot of variation. Most of the roots, almost all the species in the region are arbuscular mycorrhizal, which is one thing we can, so that's one thing we can eliminate immediately. But there's an idea, so we're very interested in how trees die, or what restricts trees, how pathogens restrict trees. And we're interested in this phenomenon of conspecific negative density dependence, which is that trees tend to die more quickly as seedlings when they're close to their parent tree. So trees of the same species suffer greater mortality close to home, if you like. And so one of the possibilities is that this varies with phosphorus. And so perhaps species at low phosphorus have different root traits, different root morphology, perhaps finer roots in search of phosphorus compared to high phosphorus, where the roots perhaps are shorter and thicker. And this perhaps confers greater susceptibility to pathogen attack at low phosphorus. So that's something we're working on at the moment, actually with a group from Canada, from Montreal, from the University of Montreal. They're studying this in some of our plots. So I think it's a great question. I suspect there's something in this, yes, that root trace will explain some of these patterns.
Okay, I think we're going to cut it off there. Take one more second just to give Ben a round of applause.